So I figured I'd start in and give you a little background information about sweet potato crop itself, and then talk about my project, and then go into how it relates to combating climate change. So the sweet potato already on its own is like one of the most important crops in the world. It's the third most important root crop in the world and the seventh most important crop in the world by metric tons. So over 100 million tons are produced annually worldwide, and it's also a strategic crop for humans because it's often thought of as a famine food. It's going to be like your one food that gets you through, even if you experience like drought or flooding, something that could devastate in a lot of other crops, which is super important because apparently that's everyone's biggest issues with climate change. We saw from the survey earlier. So, and it's one of the most versatile crops too. In addition to being used to make pies and yams for Thanksgiving or Christmas dinners, humans can consume regularly the roots and the foliage. It has a lot of nutrients, key nutrients, essential to eye health, like beta carotene and vitamin A, and the foliage can also be used to be fed to animals, and it's a common landscaping plant because it's very viney. Like the vines will grow everywhere if you don't look out. Now, one of the key things you need to know about sweet potato is it can be a no-till crop, and I would know because I planted all my potatoes and harvested with this thing here. You can see it's like a plow. It's basically a pitchfork, and you just you go out there until it's done. And you get kind of a rhythm with it. So it's great because you don't need to use those giant mechanical tractors. So you're using less fossil fuels, less energy is consumed that way. And it has an extensive root system. So a lot of people have argued it's a plant that can be used to combat erosion, not just water erosion that occurs like in wetland areas, but also wind erosion. It can be used as a cover crop in some cases. And biomass yield, it has a massive biomass yield, so it can be used potentially for plastics and fuel production. But there's challenges to creating a genetically superior crop because although sweet potato tends to be fairly resilient when it comes to like super like flooding conditions and other adverse conditions, and very heat tolerant, it is very difficult to cross. So it's almost to the point that it's sterile, and it's usually reproduced vegetatively through cuttings that are also called slips. These little guys here, you just snap one of those off and put it in the ground and it'll take off. And so any resulting seeds that do occur in the sweet potato are often not viable, and it's thought to be due to their polyploidy nature. And currently the genome is not sequenced, so there's already a ton of issues when it comes to breeding for a genetically superior crop. The key word being actually breeding, because the cultivars and accessions that are around now, they've been around and there's really no way you can variate them from season to season. So the key is to get a good one for your area. Now, my biggest questions in my project is I'm looking at gene expression primarily in my sweet potatoes. So I have 3 different genotypes. Let me go to the next slide. I have 3 different genotypes here, and I'm comparing the 3. So what I'm doing is I'm isolating mRNA from leaf tissue and preparing a library and sequencing just the RNA. So we're comparing gene expression between the 3 genotypes, and I have a yellow fleshed, a white fleshed, and a purple fleshed genotype, and they're called 7, 37, and 39 because that's their names. Those are just the accessions that I actually got from Dr. Podell, actually, who got them from Dr. Bonsai in Tuskegee University. So we're mapping. And then once I've sequenced all of my RNA, I'm mapping and carrying out fold change comparisons with CLC Bio and the transcriptome data, because we don't have a full genome for comparison and for mapping to be carried out. I'm using a transcriptome data from the Max Planck Institute in Berlin, Germany. So that's pretty cool. Like, I didn't even know there was a transcriptome. Actually, it came out last year, so I got pretty lucky with that. So what my project questions is, what is like normal gene expression? Because I don't have any symptoms. I'm not comparing like Isaac's plant was. Like, I'm not comparing, all right, drought tolerance. I'm not comparing plants that have been flooded. I'm not doing that. So I'm just looking at what is normal gene expression in fully matured sweet potato plants, these 3 different genotypes, and what are major genes involved in biomass accumulation, because as you can see, they're very different. Like 39 is like the monster of the group. Like, it takes off and there's a lot more bioaccumulation. And being that they're all the same, they're different genotypes, but they're technically all Ipomoea batatas. So they're all sweet potatoes. They're all the same, technically. So I'm looking at major genes involved in biomass accumulation, because that would be very helpful for someone trying to develop, like in the future, if someone's trying to develop, like I mentioned before, fossil fuels and plastics manufacturing. So I'm also looking at genes involved in nutritional content, starch and sugar accumulation, simply for like the human aspect of it. Like, you got to keep your people fed too while you're saving the world. And you also need to know what genes are involved in changing fibrous roots into storage roots. Like, I'm looking at genes involved in that, as well as any kind of phenotypic differences having to do mostly with yield, but like also you got to be able to have your people eat the food too. Like, if you have something purple and they have a thing against eating purple things, they want to eat your orange potato, even though the purple potatoes come out like much bigger, consumers aren't always going to want that, and you have to be aware of that too. So this is my workflow. My first thing is to extract the DNA, extract RNA, do a quality check, all the way to library preparation, and I have my steps in here. And that's library preparation is just a nice way of saying take up 3 days in a row of time. Like, it's a very strenuous process on the lab bench. But we get ready for Illumina sequencing. So I send that off for sequencing. We actually send it to University of Delaware. And so basically I get all the data back. I get all of my reads, and it's just nucleotides at this point. So my next step, which is where I kind of am right now in the CLC bioanalysis portion of it, is to import my data and align it to the Max Planck data. And so then I can annotate my reads. So every single one of those reads that I get back, I'm going to know what gene they went to, and then I'm going to be able to look at them and be like, okay, which ones were expressed more, which ones were expressed less, and which ones are expressed higher in a single sweet potato strain, or in this sweet potato strain. I can compare all 3 of them right across the board with my fold change analysis, and then I can sort genes into functional groups to make it easier to break down. Okay, where do these genes go? Like, okay, let's say you have something for B amylase. There's a gene I expressed there. It's like, okay, well, where does that fit in functionality wise? So yes, comparing my profiles across 3 different genotypes. That's actually all 3 of my genotypes in that picture. I don't remember. I think down in the corner here is 37, and then 39 up in the corner, and then down here is 7. So they all, I just wanted to show that they're all like a little different when it comes to like pigments. So we want to closely examine genes, especially when they're related to disease resistance, stress resistance, starch accumulation, and sugar accumulation. Now, the biggest thing that I wanted to investigate at first, it didn't end up working out that way, but hopefully my next step will be disease resistance. I'm going to be looking into sweet potato viruses. I actually got some accessions that were infected with viruses versus not infected with viruses. And the reason why they're a problem is, here's some of the symptoms that occur, which really okay, you have vein clearing, chlorosis, leaf strapping, leaf distortion. None of those are good for photosynthesis. So obviously, you're going to have your yield impacted. So if you were to get one sweet potato viruses, of which there's about 30 of them known, like that's not as big of a deal as if you were to get 2 of them. Whereas sweet potato chlorotic stunt virus and sweet potato feathery mottle stunt virus, they actually form a disease complex called sweet potato virus diseases. They're so creative in naming that, but that's just what it's called. The issue there is this is actually the disease, virus disease has been known to cause up to 100% yield losses. They go to dig up their potatoes, there's nothing there. Like, you may have had a lot of biomass accumulation, but there's no potatoes. So obviously that's a problem when you've done all that work and put all that energy in to not have a yield. So this is where my collaborative work with Arkansas Pine Bluff comes in. I was going to look, as sweet potato viruses accumulate generation to generation, I was going to look at, all right, generation 1, like just cultured, and then the first filial generation, season 1, season 2, and season 3, they accumulate so well because you're going to have cuttings, and that's going to grow into a potato. You're going to harvest the potato, and from that you'll get cuttings from next year. You haven't gone to seed because, like I said before, they tend to be a very sterile crop. So yes, in doing this, I was going to use reverse transcriptase Pcr and quantify the copies of viruses, because obviously copy number of viruses will go up from year to year. And theoretically, you would see, as in most cases, you'll see a decrease in yield as it goes along. And I want to see what genes are upregulated versus downregulated. You're like, well, how is that useful just to see that your yield is going to go down? Well, I want to see what genes are upregulated, and if any of my 3 cultivars are more resistant as a result of an upregulation or a downregulation of their genes. So we really don't know what we're going to see yet. I have the samples, actually they were sent to me. So hopefully if I come back again, I'll be presenting that data to you. But the impact here, the major impact is I wanted to find genes related to the most important phenotypic qualities like starch accumulation, biomass accumulation, and of course root differentiation. And this way I'll have a reference genome for future work, like I mentioned, like with the University of Arkansas. And I also want to give, like we've been talking about all day, give farmers an edge when planning their crop for the season. And I want to make them, like, we all want to make sweet potatoes a much more favorable crop, especially in like isolated regions. And our connection to climate changes, to reiterate, they're commonly a no-till, and they can prevent water and wind erosion. And you have a large source of biomass for plastics and fuel production. And they're tolerant to many adverse conditions, and they are highly nutritious in addition to all that. Yes, picture.